And I am your forensic analyst, Bea Jamel Pastor. That's right. So, our job for today is to understand the crime, the genetic diseases, and learn about the most advanced tool in our arsenal to fix it. So, every one of you in this room is a vital witness in cracking this case. Okay, but before that, let us first know the learning outcomes. So, by the end of this briefing, you, the witnesses, will be able to first define what a gene is. Another, differentiate gene therapy from stem cell therapy. Explain the importance of gene therapy in the future. Demonstrate processes of gene therapy, and finally, show in life situations genes' importance in education. Alright, so that brings us up to our ultimate weapon, the gene therapy. Okay, so what exactly a gene therapy is? Okay, a gene therapy involves altering the genes inside our body cells in an effort to treat or stop the diseases. Okay, so in other words, or basically, gene therapy involves changing the genetic codes in order for a person to, or in order for the scientist to stop or treat the diseases. Okay, so um, this gene therapy is a medical treatment that uses genes to stop or prevent diseases. Okay, so let's say for example the liver congenital amaurosis. Okay, so this liver congenital amaurosis is it is a disease in a form of inherited blindness. Okay, so a patient who were legally blind can now see after a single injection of a corrected gene, so directly into their retina. So this isn't science fiction. This is happening right now. Okay, so sibal man mo. Okay, moving on to the genetic engineering. Okay, so what is genetic engineering? Okay, so a genetic engineering involves removal of a desirable gene from a gene, adding it to the gene structure of a bacterial cell, replacing the recombinant DNA into the bacterial cell. So the bacteria then have the capability to produce the protein by the original animal or plant cell. Okay, so basically genetic engineering, so this is a process or a method wherein it's like a specific concept for gene therapy. Okay, so it is a method, a process wherein a single gene or the disease or affected gene is removed or replaced by a healthy gene in order for a person to stop or survive. Okay, so through the powerful technique of this genetic engineering, we have the potential to not just solve these cases but also prevent them from happening. Okay, so let's say for example in medicine. Okay, so uh, let's say the production of synthetic insulin. Okay, so this production of synthetic insulin is a classic win for genetic engineering. Okay, why? Because instead of relying insulin from pigs, they inserted human gene for insulin into a bacteria. Okay, so this bacteria became tiny factories producing pure human insulin for diabetics worldwide. Okay, this shows the ability to reprogram organisms to solve medical problems. Okay, so do you know that the first medical or first synthetic insulin produced was using E. coli bacteria? Okay, so in 1978, and was first sold commercially in 1982 under the brand name Homulin by Eli Lilly and Company. Okay, so for you to elaborate more or understand more, so let us say, for example, let us add another example. So let's say in research. Okay, so scientists are using tool called the CRISPR-Cas9. Okay, so this CRISPR-Cas9 is a precise gene editing technology to create genetically modified animal models of human diseases. Okay, so it allows them to study how diseases like Alzheimer's progress and to test new drugs in uncontrolled environment to help them accelerate the discovery. Okay, so imagine that is how uh, amazing the discoveries of the scientists nowadays in terms of creating or solving the problems for our health. And do not forget what we're ultimately fighting: the gene mutation. Okay, so let's move on to the gene mutation. Okay, so what is a gene mutation? Okay, so a gene mutation is a permanent change in a DNA sequence that makes up a gene. Okay, so these changes can be caused by errors during DNA replication, exposure to environmental factors like UV radiation or other types of DNA damage. Okay, so basically, this gene mutation is the root cause of the corrupted code we see in our case files. Okay, so this gene mutation can result in a different proteins being produced, so which may alter an organism's traits or cause diseases. Okay, so basically, this gene mutation is the root cause or the problem or the main problem in this case. Okay, so let's say for example the hereditary breast cancer. Okay, so this hereditary breast cancer can be acquired through genes. Okay, but most cases are not directly inherited or caused by inherited genes. Okay, so this BRCA1 and the BRCA2 are the most common genes we see in hereditary breast cancer. Okay, so this BRCA1 and the BRCA2 normally help repair damaged DNA. Okay, so when mutated, they become less effective uh, and has a higher chance of cancer. Okay. Okay, moving on to the DNA or the three nucleotide subunits. So they are the phosphate group, the deoxyribose, and the nitrogenous base or the nitrogen base. Okay, uh, so before we dive into the three group or three subunits, so let us first recall what a gene is. Okay, so a gene is, in simpler terms, are the basic unit of heredity that can be passed from a parent to their child. Okay, so a gene is composed of subunits called the nucleotides. So this nucleotide or each nucleotide is composed of three subunits or 3 smaller subunits. So as what we have mentioned earlier, the phosphate group, the deoxyribose, and the nitrogenous base or nitrogen bases. Okay, so what is a phosphate, or what composes of a phosphate group? So a phosphate group is a group of atoms that is negatively charged and form the backbones of the DNA. Okay, so this phosphate group is a chemical group that made up of phosphorus and oxygen. Okay, so another is the deoxyribose. Okay, so this deoxyribose are covalently linked to phosphate group, also contributing to the backbone of the DNA. Okay, so this deoxyribose, ribose rather, is a 5-carbon sugar that made up of carbon, oxygen, and hydrogen. Okay, so now moving on to the last group, which is the nitrogen base or the nitrogenous bases. Okay, so this nitrogen base is a chemical unit composed of carbon, oxygen, hydrogen, and nitrogen. Okay, so this nitrogenous bases has 4 basic or 4 main base: the adenine, the thymine, the guanine, and the cytosine. Okay, so these 4 specific or 4 bases are the specific sequence for these bases that encodes genetic information. So they can be arranged depending on the gene's sequence. So this contains instructions for making a protein. Okay. So, for example, the A or the adenine always pairs with T or thymine, and the cytosine always pairs with the guanine. Okay.